I'm going to talk today about some a problem that I've been working on lately, and I think it might be a good thing to consider for all of you guys, because I think I came at it kind of broadly in terms of about principles for biology. As Mara mentioned, let's see.
You want me to move this thing?
Okay, there we go. My lab uses different approaches, both approaches from the informatic as well as the axiomatic domain. So developing interpretable deep learning methods where we can understand to distill patterns of data into human understandable code. We use molecular simulations to understand protein dynamics. And we also use some tools of statistical mechanics to try to understand if there are some general principles to be had. And kind of the question that my lab really tries to think about or focus on is, are there some principles of biomolecular information encoding? So what do I mean by that? So, well, we work on problems in terms of how a sequence, say, of a protein encodes its function, how signaling networks encode the information from upstream to downstream, how information propagates through the physical medium of a protein, like what is the material property of a protein to go from one part of the protein to the other in allostery, and how information is encoded in the aggregation or self-assembly of proteins. But today I'm going to talk about the relation between information and the energy gradient that is provided in the cell. And this really is the work of a very talented student, Ethan, who's been working for a long time. And this is the culmination of a lot of years of us kind of sitting and thinking and going back and forth. Okay, so we know that biomolecules perform the essential functions that are the basis of life. And in a sense, we can think of biomolecules as performing non-equilibrium computation, right? So here's 4 categories of what biomolecules do. They transport things. They obviously are involved in replicating at the molecular level and therefore at the cellular level. They're responsible for providing the infrastructure through self-assembly, right, of actin and microtubules and other large-scale structures inside the cell. And of course, they're involved in communicating information from outside to inside, and they do very complicated combinatorial information processing to really decide what they should do. Right, and all of these involve the conformational, configurational dynamics of proteins. They fluctuate and they transition from one state to another, and they're also doing this in a state in which they're typically very, very far from equilibrium. Right, so that makes sense for processes like transport, where if you're at equilibrium, then of course every forward reaction is matched by the reverse, and so you cannot have any net transport, and of course you can't have replication. That all makes sense. But if we think about other kinds of aspects of biology, you can actually think of, well, maybe biology could have chosen through evolution to just do computing using equilibrium paradigm, right? So there's no reason why biology couldn't use just equilibrium self-assembly. But in fact, actin and microtubules, they actually involve hydrolysis of ATP and GTP to build themselves up. And of course, in the signaling realm, most of the important signaling processes in the cell actually consume energy through things like post-translational modifications like phosphorylation, right? So, but you can imagine, in principle, you could have an equilibrium model of computation. But just like in these processes are typically very far from equilibrium too. And kind of the question I want to talk about with you guys today is, like, why do we need that? Like, what do we get from that, right? So why doesn't nature just always use cheap equilibrium computation? So just to make that concrete, let's just consider like a very simple 3-state model, right, of what it would look like for equilibrium computation, right? So imagine that there's some protein A, and it can bind to, say, another protein B, and some other protein that's called the receptor R, right? So here it's in a free state, they're not bound. Here A is being bound to B, and here, because A is bound to B, it can now perfectly fit R, and all three are bound together. And let's say that this complex is the activating complex for the downstream signal. So however much of this system is in this state determines how on the signal is. Yeah, so here's an example of equilibrium computation in the sense that if I, for example, change the amount of B or change the binding affinity from B to A, I can modulate how much of the system is in this on state, right? And this would be equilibrium computation. I would maybe spend some energy in producing or degrading A or B, but once it's set at that steady state, I don't have to put in any more energy. Yes, go ahead.
What do you mean by computation? Like, I don't see a NOT gate or anything like that
Well,
here.
Yeah, so that's a good question. I'm gonna actually talk about it a little bit more in detail, but computation is really, let's think of it just very simplistically as there's some input, and let's say the input leads to downregulation of how much B is produced. And because of that, there's less B, and therefore I shift the equilibrium away from this state towards this state or this state. So that would be a computation that I have some upstream thing. It tells me degrade B, and because of that, there's less of this state, and this state is the starting signaling state for the next step of the cascade. Right. Okay. Oh, sorry, something.
No, it was just
Just hiding the panel.
Okay. So, but what actually happens in most cases is something like this. Also, just for apples to apples, you can think about as a three-stage process of non-equilibrium computation, right? So now let's imagine there's again A, right? But now through a process of phosphorylation, where another enzyme called the kinase, right, utilizes one unit of energy, it hydrolyzes ATP, right? And actually takes the phosphate group and attaches to A. So this is a phosphorylation step. And then because of it's phosphorylated, then it's enabled to bind to the receptor R. And now this process is driven in the forward direction because there's going to be a net, production from ATP to ADP. Okay, so even though this process can in principle happen in reverse, it happens with like way less frequency than before. So there's now it breaks what's called detailed balance, right? So in the equilibrium case, the flux here is exactly balanced by the reverse flux, but in this case, that's not true. This flux is greater than this, and this flux is greater than this, and this flux is greater than this, and it will lead to a cycling between the states. So it's still at a steady state, where the probability of being in this state or this state does not change in time, but it's in a non-equilibrium steady state because it doesn't obey the detail balance, right? It's cycling through, and to maintain that steady state, it's constantly burning ATP. So now I can also do computation, and biology does do this form of computation in its phosphorylation cascade, right? Now when I say change the amount of the activity of the kinase, right? I can modulate how much phosphorylation happens, and then I can modulate how much is in this on state, right? So I can kind of just spell that out in terms of like what that would entail, right? So now let's imagine the three different states that I've drawn here: A complex with B, right? Or A complex B complex with R, which is the on state, right? So I can imagine at some initial configuration, here's an equilibrium probability distribution. Now I produce B, I produce more copies of B, and I shift the steady state equilibrium probability to this, right? And now I've upregulated the on state from a tiny bit to this much. And I can do another step of computation. I produce even more B, I reach another equilibrium steady state. I turn it on even more, and then maybe I degrade B and then I turn it off, okay? Now, in every single one of these computational steps, the y-axis here is I'm plotting the power consumed. And so what's happening here is that at each one of these computational steps, I have to use energy to produce or degrade B. And so there's some transient power that is being dissipated or consumed to do that. So this would be like you can think of like as an energy cost, right? You only pay the energy cost when you're actually doing the computational steps themselves. In between, you can have a long period of time where you want to keep it at whatever state it is, and you don't have to pay any energy, you just leave it at that equilibrium state. So in contrast, the phosphorylation paradigm that I talk about, where you're making or you can do the same thing, right? You can turn on and off this on state, this APR state, right? At each step, you're say producing or degrading copies of the kinase. So just like in the previous example, you're consuming energy at the steps of computation, denoted by these little horns here. But importantly, even when no computation is being performed, just to maintain the steady state cycling, steady state, you are always burning through ATP. So that's this solid area under the curve here. So the question more concretely of why doesn't nature always use cheap equilibrium computation is why doesn't nature just do this? Why does it do that? To ask it kind of more precisely, then we can think, well, there could be many, many different reasons why actually. And maybe what I'm talking about is just one of many reasons. And maybe in some cases, it's not the biologically dominant reason, but I'm going to focus on what computational advantages is gained from doing this versus that. What computational advantage is gained from actually applying a thermodynamic force, right? In this case, driving the cycle, this net cycling that we can get. And so what computational advantage do we get from the energy that is consumed? Okay, so to kind of answer that question, we have to like just define precisely what we mean by computational advantage and thermodynamic force. So in the next few slides, I'm just going to define what those things are, and then we can get into it. Yeah, are we all good so far? So good. At least with the premise of what I'm saying. Yeah. Okay. So how do we quantify thermodynamic force? So let's imagine here is the state space of some complicated molecular system. It can hop from one state to another, etc. And when I first draw this black state space, right? Imagine that is the reference equilibrium system of that system of whatever molecular machine or system I'm thinking about. So that is the system in the absence of coupling it to any, say, ATP gradient.
Can
Okay,
I
go
actually
ahead.
relate it, as you said, sort of set up for the beginning part? I think there's sort of two things in my mind. One, from a modeling point of view, even if you're modeling, but not sort of biophysically, not physically, you make a bunch of hill things, a bunch of switches. It seems like, you know, applied mathematics thrown at signaling cascades. You can model everything and you can pretty much get anything you want out, any input-output response. There's no mystery.
Yeah.
So one version of the question is, what's the value of grounding that in energy gradient? What's the additional value in that way of thinking about modeling? And secondly, if you go into biology, energy, no one thinks about energy. It's just irrelevant to how cells are evolving or computing or anything. Yeah, just your thoughts on both of those things.
Yeah, well, I think, let me answer your second one first. I think one of the questions to ask is like, why is it relevant? Right. So like we know that we know what the energy budget is for the, I mean, most of it goes to just like making ribosomes, right? But the question could turn out to be that in order to do the computations on top of that, the amount of energy is like way, way more, right? So the one way to frame your question is actually why is the energy expenditure of the cell in ribosomes and not through maintaining these fluxes. So the answer to that would be quantitatively, because we can get a lot of computation for a reasonably modest energy budget. Yeah. So I think it's more like, well, why is the case that is, right? I think the other kind of thing is like, is that, you know, if we want to understand certain things, sometimes one of the things we want to actually be able to do is actually build these systems like completely orthogonal to the building blocks of what nature uses. Like it would be nice to be able to build nanoscale machines that does the type of input-output dependent computation at the molecular level that nature has achieved. And to do that, I think we also need just basic principles of like, well, under certain conditions without a certain amount of energy gradient, for example, maybe certain things are not possible. I mean, I'm not going to talk about this work here, but one of the things we showed was that if you want to make a chemical engine efficient, right? So efficiently use energy, you have to have a threshold thermodynamic gradient. Otherwise, it will actually inefficiently use whatever energy you provided. And that efficiency goes down to almost zero. So it's not like, oh, you can kind of eke by. It's like there's like a sigmoidal switch, right? So kind of these design principles, I think, are important. Well, can you remind me what your first question is? I think I had a two-part.
No, you answered. It was the first one was applied math modeling.
Okay.
Where you just sort of say, you know,
Okay.
dynamical systems.
Okay. All right. So so let's imagine this the reference equilibrium system as the system before I've coupled it to any thermodynamic gradient. So in a case where I couple it to a system where ATP is, let's say, in equilibrium with ADP, so there's no net hydrolysis of ATP. Okay, that system would reach equilibrium, and that's what I denote by this just black network. But now I'm coupling the system to energy gradient. I'm coupling it with hydrolysis of ATP. And so what the way I'm going to denote that is I'm going to draw these red arrows. Okay. So every time I draw a red arrow, that means that this is an ATP, let's say an ATP binding step. So normally it would reversibly bind and reversibly go from something that's not bound to ATP to bound to ATP and vice versa. And now I'm going to dramatically increase the rate constant of binding ATP, okay? Because I'm actually actively injecting ATP into the system to maintain a really artificially high balance of ATP to ADP, and that's what's fueling this whole thing. So that's what these three red arrows are. And if you actually zoom in on what that looks like, you know, locally, there's all these forward and backwards rate constants. And what I'm adding here is just an additional, what I call alpha rate constant of binding to ATP. So the applied thermodynamic force then of this forward reaction is just kT times log of 1 plus the additional non-equilibrium rate constant divided by its original equilibrium reference rate constant. Okay, so you notice that that means that this theta ij, this applied thermodynamic force, is only non-zero at the wherever the red arrows are. Everywhere else, the alpha ij is zero, and there's no applied thermodynamic force.
Why is it that you're focusing on the binding and not the unbinding or the step at which you go from ATP to ADP, right? I mean,
Yeah.
it's really just the cycles that you have.
Correct. Yeah, so this is all defined in terms of a reference system. So you can always define your reference system to be a fixed ADP concentration. So that means you can always choose a reference system in which like all the red arrows, just for mathematical convenience of solving it, is shuttled into the binding step. Right. So, but yeah, like you can choose any reference equilibrium system you want. All of the results, as you see here, are always going to be defined in terms of a given reference system. So there's no ambiguity about like, what am I comparing it to? Okay, and regarding to Todd's other question, so we show that at steady state, the total rate of dissipation is a sum over all the edges of the net current, which is I Mn times this applied thermodynamic force. So that means that the terms in the sum that are only non-zero when it's theta Mn is non-zero. Okay, so even though entropy is produced everywhere, even though dissipation is happening everywhere at every single transition, right? When you push the system out of equilibrium, to compute the total dissipation, you only need to add up the contributions at only the edges that you drive. If you sum along any loop of this applied thermodynamic force, that is going to be equal to the total, the sum of the chemical potential difference along that cycle, which is called the cycle affinity. So the cycle affinity, which is the sum of the chemical potential difference around the loop, oftentimes is used as a measure of thermodynamic force. Here, we're going to use a different viewpoint, where we're gonna look at the applied thermodynamic force theta, but it's still related to that in the sense that if you were to sum this theta along any cycle, it's the same as the chemical potential difference along that cycle. But the difference is that the theta ij is only a function of directly tunable parameters, like your rate constants. Whereas the chemical potential difference is like a collective property of forward and reverse fluxes summed along that. Go ahead.
So how would your force relate to the thermodynamic affinity?
Say that one more time.
How would your definition of the force relates to the thermodynamic affinity? So with the chemical potential difference between the reactant and the product of that specific reaction.
Yeah, so like you're talking about the specific reaction, like so with every single edge, there's a reaction, right? So that means if you sum the change in chemical potential around this edge and this edge, this edge, this edge, this edge, this edge.
No, yeah,
this edge,
that I understand. I'm asking the sigma ij that you define as KT logarithm of 1 plus the ratio.
Yes,
You said that that is a measure of the thermodynamic force.
yes,
And the thermodynamic force along a single step, single
Yes.
reaction, should be the difference between chemical potential gradient between the reactant and the product of that reaction. My question was how that quantity relates to this.
yeah. So it doesn't relate. It's not related to the direct affinity or the chemical potential difference along that edge. It's only related to it if you sum around the whole cycle.
Okay. So let's say it's an alternative definition that coincides with the standard one over cycle.
Yeah.
But for a single reaction,
Yeah.
it's something
It's
different.
something different, yeah. And the difference is that it's only a property of the locally applied parameters, right? Whereas the delta mu is actually the log ratio of the forward and reverse fluxes, which is a collective property. But also another way to think about that is, like we can think of this power, the dissipation power, right, as a velocity times like an effective force, right? So now we're just thinking about of applied force rather than right. So okay, so and then I'm going to define the total applied thermodynamic force, right, as the maximum along all possible non-intersecting paths of the sum of these little thetas, these forces. Okay, so that means in this particular example, the best I can do is I can't actually get I cannot choose any path where I can get 3 of these red arrows, right? Like for example, if I choose this path and go like this and catch 2 of the red arrows, but I cannot catch this third one. Like if I want to get this third one, right, I could do something like this, catch that third one, but then I can't get this one. So in this case, the capital theta, right, is either this red arrow plus this red arrow, or this red arrow plus this red arrow, or this red arrow plus this red arrow. Capital theta cannot be the sum of all 3 red arrows. Okay, so the capital theta is a measure of kind of the total thermodynamic force, and it's a synonymous with how using the affinity measure, the total affinity is usually characterized as what's the maximum of the sum of delta mu along any cycle. Okay, so that's the quantification of applied thermodynamic force, like how you're going to drive the thing. And then here is the how we're going to quantify what it means to compute something. Okay, so let's say the computation is just transitions between steady state probability distributions. That's the model that we're using now. And there's going to be discrete transitions. We're gonna think about, so we'll have a system in some steady state, we'll have a steady state probability distribution, and that system is parameterized by a vector of a bunch of parameters that determine that steady state. I just call it K prime. And then what I'm gonna do is when I perform a computational step, I'm gonna change some of these parameters in K prime to other parameters, and I'm gonna call a new vector of parameters K. So the computational step is what happens to the probability distribution when I change K prime to K. And so
Sorry, K and K prime are parameters of the distribution
the parameters
of the generative process.
yeah. So rate constants, concentration of, my previous example could be concentration of that protein B. It could be a mutation. I mean, you can think about it as even a mutation. I mutate a protein and now it's binding affinity to something different. All of these are the elementary parameters. So I'm considering any arbitrary change in the elementary parameters of the system such that it now gives a different steady state distribution, okay? For biology, I think the two things that we're going to be interested in is we're going to be interested in first, we're going to always fix the applied thermodynamic force theta. So because usually in biology, the energy gradients that you're harnessing, like the ATP gradient, is fixed, and what you're changing is like, you know, binding affinities, phosphorylation, the parameters of your molecules, but the harnessing of the energy is fixed. Okay, so we're gonna have a fixed theta, but we're gonna change K prime to K, right? So one of the things you might be interested, if you're, for example, in a signaling protein, you might just be interested in, let's say, I is the signaling state, the state that is important for downstream signaling. We're interested in, initially, when my system was had parameters K prime, what is the probability of being in that signaling state? Now I change K prime to K, which could be many different things. I can do lots of things about the system. What is my new probability of being in the signaling state? That would be amplifying the probability of a particular state. This would be relevant for the signaling application I gave. Alternatively, you might be interested in actually in how the entire probability distribution changes. So sometimes you're interested in just like, if I go from K to K prime, not just how much does this little signaling state gets amplified, but how much does the entire probability distribution change, right? And in that case, we're interested in the KL divergence between these two distributions, right? Which I'm going to devote this. So KL divergence in the presence of a fixed thermodynamic applied driving force between the distribution of K prime to the distribution of K, right? And if you know the probability distributions, you can calculate the KL divergence, which gives you a measure of the differences between the post-computational distribution relative to the pre-computational distribution.
Just want to understand capital theta. If you fix capital theta, is that like setting ATP to ADP concentration fixed for the graph of reactions?
Well, it's like setting. It's a
global thing, no?
Yeah. So you can have many reactions in your system. Some of them could be ATP to ADP. Some of them can be like a proton pump. Right? All those like red arrows, the capital theta just means that you're not messing with any of your red arrows. Okay. So it doesn't have to just be ATP. It could just be whatever is driving your system. Let's assume that what's driving your system is being fixed, and then the system parameters themselves are being charged. It's kind of like, you know, when we're plugging something into the wall, we assume that like the voltage in the wall is being fixed, and then our computer or our device is changing. Right? And that's kind of how the cell to first order operates. It maintains these gradients that it maintains are pretty constant. Okay, so these are the two questions. These are the two things we like. So then formulated it now more precisely, you know, the original question, why does nature use cheap equilibrium computation, is really how did this thing, which is how does an individual state get amplified, or this thing, how does the entire probability distribution get changed, depending on how much the system is being driven, like how much voltage is in your wall. Okay. So that's actually just the question of my whole talk. So that's what I'm going to talk about. Okay. Are we good with? And I know that, you know, this is to make it concrete, we had to define it like this. There's obviously, you can think about, well, what if you compute continuously and not in terms of a discrete jump? But just for concreteness, this is kind of the problem that I'm posing. Yeah. Go ahead, yeah.
Why did you define theta as the maximum over all the driving forces and not just the sum over them?
Oh, because in some cases, the sum over them is huge. Yeah, so you can imagine something where you have lots and lots of parallel paths. Imagine you can go through this path, or you can go through that path, and they're all parallel paths with just slight differences. You don't want to be summing over it, because then I could just come up with another system that just has like a few more alternative paths and they're all driven. And now my theta is suddenly much bigger. I see. Yeah. And the result that I'm going to talk about then would also not be tight. Okay, so let's go back to the three-state system because it's easy to like visualize first. So, you know, the way that we're gonna think about this is that here's it's hopping between different states. We're going to think about this in terms of the rate constants, right, that I just talked about, the equilibrium rate constants, and then there's gonna be the theta, which is, I'm not gonna change, right? The theta comes from driving certain rate constants. Additionally, and for convenience, I'm going to parameterize this using like a circuit parameterization where you can write this equivalently as a Ohm's law relation on a circuit. And so all the things that involve these passive equilibrium parameters go into like the free energies. These are the free energies of the reference equilibrium system, and then the resistors. Right, and then there's the theta that we talked about, which is which corresponds to a battery. This parameterization allows us to solve the things that we want very efficiently. And what we can do is using this parameterization, we can numerically investigate this problem. So what we take is we just take this three-state system and we just generate a whole bunch of different numerical examples and see what happens. So what this plot is showing is that, okay, if I now generate a bunch of different examples of the three-state system where I can mess around with the free energies, I can mess around with the resistors, but I'm always keeping this theta fixed, right? So now as a function of what this theta is, I'm asking how much information gain can I have upon a computational change? And here's the value if theta is 0, so I can have equilibrium computation as expected. Right, at equilibrium, I can mess around with the probability distributions, but as I now drive the system further from equilibrium, you can see that for the same exact system, right? The fact that it just has been driven in the background of being driven, you can actually have a much higher information gain once you change your parameters. And these four curves just tell you the number of parameters I'm allowed to change between k and k prime. So of course, if I only allowed to change one parameter, I can't get the probability distribution to vary too much. If I'm allowed to change 4 parameters, I can get the probability distribution to change more. But the main thing is that no matter how many parameters I'm allowed to change from k to k prime, if it's in the context of being driven, I can always, in this numerical example, I can always get more information gain. So the KL divergence is always more. So I mean, this is the numerical result. So what we would like to understand is can we understand this more generally and as a more general constraint of molecular computation, right? So here I'm asking the question again: how does this or this depend on the total applied thermodynamic force? And what we would like to find is an answer that is mathematically concise, so not just like simulations that I showed before, but we also want it to be like tight, so it's actually constraining and not like some really huge, like ridiculous bound that, of course, nothing's gonna, that's obvious or nothing's going to disobey. We also want it to apply to any system parameterized by any set of, right? Right. And we also like to apply it to any computational step, so we can change whatever we want, any arbitrary computational step. And we also want it to apply to the background of any theta, including multiple transitions that are driven far from equilibrium. So that's the result that we would like to find.
Ask a quick question.
Yes.
When you measure your information gain, like, why is it always good to have a larger information gain? Like, is there not like, I could imagine that I'm just going to generate a ton of heat in my system, right? Between two distributions, because like there is some cost to these changes. So why is it always better to have
High?
Yeah.
Now you can also imagine cases where I framed it as computation where I go from k to k prime, these parameters are changing, right? As a purposeful thing, like the cell has decided to go from k to k prime based on some input signal. But you can imagine stochastically, k just goes to k prime due to like copy numbers changing and some things that didn't anticipate. And in those cases, you might not want information gain to be like, you actually want to minimize the information. You want to preserve the probability distribution, right?
Yeah.
Yeah. So we'll get to that. Yeah. The bound that I'll give will actually be a bound both for amplifying the information gain as well as suppressing how much you can suppress the information gain. So it turns out that if you drive it, you can also suppress the information gain too. Okay. So the thing I'm going to go through is just show you the proving. The first thing we're going to do this systematically. So first we're going to prove that the probability application of a single state now with k fixed and variable theta. So if I have the same system, but I change my theta, how much can I amplify that? And remember, that's not the original question, okay? But I'm going to use that to get to the thing I care about. So then I'm going to use that to ask what is the probability application of a single state when you change from k prime to k for fixed theta. And I'm also going to use it to answer the question of how does the KL divergence change over the entire probability distribution if you go from k prime to k, also for fixed theta. And then I'm going to show you some numerical investigations into how tight this bound is and give you some biological examples. So that's the roadmap for the rest of the talk. Are we still cool? Okay. All right. So let's do some more numerical examples. And I find, I think Madhav agrees with me on this, that just playing around with stuff often gives a lot of good intuition and can actually constrain how you might want to mathematically tackle a problem. So let's now consider, Ethan actually generated like 100,000 randomly generated networks, okay? And with various parameters in it. And the only thing is that he added 4 driven transitions, so 4 little thetas, and for simplicity, he made the 4 thetas the same. So the 4 red arrows can be added to any one of these networks, okay? And he just asked the question, like, when the red arrows are not there, what is the probability of some state, which I call PI star, this equilibrium? And then when I apply the red arrows, what is the new probability of that state? And that is the scatter plot across like the 100,000 different systems. So you can see that there's some striations here, right? So here's the identity line, and there's these sectors or lines where I've added additional dashed lines to like make it a little easier to see by the eye. And what the lines correspond to is that like for example, this dashed line is the original equilibrium probability. Where did it go? Equilibrium probability times e to the little theta OKT. And then the second line, this line, is the original equilibrium probability times e to the 2 theta OKT. And the third line is e to 3 theta OKT. And then the fourth line, this line, is e to the original equilibrium probability times e to the 4 theta OKT. And again, the capital theta in this case, that's the maximum achievable capital theta, right, is for little theta, right? And going back to the original question or initial question, you can also see that there's a symmetrical behavior on how much you can suppress the equilibrium probability if the thing were driven, right? So here, in addition to amplifying a state, you can also suppress the probability of a state, and the suppression is symmetrical to the amplification.
The quantization is coming up from the fact that the 4 arrows have the same 4 thetas.
Correct.
Right?
Correct.
So that's not fundamental in some senses. It's more the fact that the distribution can be parameterized by those thetas.
Yeah. I think the fact that the 4 little thetas are the same means that the spacing between these dashed lines are the same, but the discreteness comes from I guess it's come from the result. Yeah, go ahead.
There seems to be a larger density on the suppression side than the amplification side. Is that a coincidence?
I have no idea. I don't know. Like we just sampled a bunch of systems. I don't know. I don't know why this is denser. It could be the kinds of systems we're investigating. It could be how we systematically change the parameters. I don't know if this is due to our numerical approach or due to something. Okay, so all right, so so the first thing if we're going to first prove the probability application limit of a single state with k fixed and variable theta. So we're asking for the same for a system. If I were to drive the system, how much more can I amplify some state, right? And I'm going to use the circuit mapping I talked to you about, as well as combining it with what's called the matrix tree theorem, which allows us to calculate the steady state probabilities of these master equation systems. Okay, and so the upshot of that is that basically, if you have some graph where you have transitions between states, you call this the original graph G, right? You can from that graph you can generate what's called these maximum spanning trees that are these acyclic graphs that contain all the nodes of the original graph. Okay, and I'm going to parameterize those by the number of mu. So mu goes from A to H in this case, and then for each mu, I can choose a root of this tree, which now I call the substance. So here, I mu is E because I chose this spanning tree, so mu is E, and then I have a subscript 3 because it's rooted at this node 3. Okay, and there's actually for any G, there's like a super exponential number of these things. So it's like it's not a practical way to calculate steady state. Okay, so you do not want to actually use this guy. But what's nice about the matrix tree theorem is that the actual steady state probability of any state I is this ratio where the SI is the sum over all possible spanning trees, right? Of the product of basically the rate constants from all the states to the root of that tree. Okay, so go ahead.
So I'm familiar with this method, like in the deterministic regime, and there it applies only to unimolecular networks, let's say linear regression. Is this the case also here? So your treatment?
Yeah, so here I'm only talking about like Markov systems, like describable by a master grid. So these in principle are, if you feel like that is unimolecular, right? So all these examples, I mean, in principle, you can think about like multi-molecular things too, but now your state space will get really big.
And
right?
did you just did you look at that at least numerically, nonlinear systems, or you for the moment you're just focused to linear systems?
Yeah, just for the moment, I'm looking at just systems describable by a state space with described by just linear rate constants. Yeah. Okay, so
Those are two questions. One, I've even done these kind of calculations. Two things. One, I never have intuition for the origin of the theorem. And secondly, your intuition as to why the eventual algebraic form of the answer is often quite simple despite the complexity of the sound.
okay, yeah. So I'll give you a little bit of intuition on that just for how we, so here's the answer. Okay, so this is the answer. So if you have a system where the equilibrium probability of any state I is pi star, and now you apply thermodynamic driving forces such that the total applied driving force is capital theta, the amount that you can amplify pi is at most this. And the amount that you can suppress pi is at most this. So the intuition for how this thing that has so many things in it can reduce to a simple thing is that, so there's like two ingredients, I guess we can think about and how we approach the problem. The first ingredient is decomposing. So this is true for like arbitrarily describing systems just in terms of rate constants, okay? Here we chose to decompose it into a reference equilibrium system that has thetas driven on top of that. And we're able to then do a lot of, we're actually able to we're actually able to factor out that
In that sum,
where actually
there's the
factor out,
equilibrium portion.
we can factor out equilibrium portion that turns out to be pi star. It turns out to be like basically a partition function. Okay. The other thing is that the intuition here is that what happens when you do when you look at this is that you can think of it in terms of, okay, so where did it go? So Where'd it go? So, okay, what's actually the even more hairy in this thing is not just the numerator, but the denominator. Okay, the denominator has like a bunch of stuff because the denominator is sum of all possible. So the thing to note that helped to simplify is that when we consider a ratio of things between something in the numerator and denominator, it's a ratio between two trees. And so if you have two trees, right? Now, the denominator will have things that are rooted at different indices. So now you can think about if you have a ratio between two trees, one tree is rooted at edge I and the other tree is rooted at edge J, but they're coming from the same spanning tree, they're just rooted differently, then the ratio of those trees is just a product on the edges connecting I and J. All the other parts of the tree cancel out because they're pointing in the same direction. So those two kind of things, but I know I'm kind of waving my hand, but like those are like kind of two things that were important to like finesse this thing. Yeah. Previous work has, so like, you know, the blowtorch theorem, etc., have used matrix tree theorems to get cool results. Those tended to work in the sense that they're asking what is the probability of some state I and some state J in the same system. Right. And when you do that, you can ignore the denominator, right? Here we're asking what is the probability I, the same probability I, but now in a driven case, so we have to like dive into the denominator as well. Okay, so just going back to here, then now it makes sense why we have all these things, right? So actually, why we get the striations is that for some topology, so let's just zoom in on one network we looked at. For some, so imagine we put the 4 arrows like this, the capital theta is just 1 theta, right? Because there's like any path you choose will have at most 1 arrow pointing in the direction you want, like, right? So any other paths will have 2 arrows pointing in the opposite direction that they're going to cancel out. So here the capital theta is 1 theta. If you happen to arrange your red arrows like this, your capital theta is 2 theta. Here your capital theta is 3 theta. Here your capital theta is 4 theta. So just because of that, there's just topologically like going to be these 4 striations based on how you put your arrows. And the same goes with the suppression, because I just showed you that the bound is for amplification and suppression. So that goes for all of these dotted lines.
Yeah, it does seem like you have some preference for like really being on the limit. Like,
Yeah.
Did you have any sense of why that is?
Nah, I mean, I guess it's hard. I mean, here, maybe here, I don't know. It's hard to say. Like here, it's hard to say. Maybe here, we haven't.
I think you can mainly see it where it curves. You know, you break
Yeah,
out the
yeah,
line at
where
the top,
it
and you can
deviates from
see
it,
that
yeah.
band, it is a little banded. But
Yeah, I'm not sure.
So sorry, this is probably I missed. So PI is a generic state or
Any state,
Any state.
any state, yeah. PI star is the probability of that state when the red arrows are not applied, and PI is the probability of that state when the red arrows are applied. Okay, so now one of the things this also means is that it's very different if you engineer a thing to drive your things in series or in parallel. Okay, and this is something that was very counterintuitive to us, because you can imagine, like, I was like, okay, if I have 4 arrows and I'm all squeezing this one state, I should be able to get that state to like really pop, right? I have 4 arrows all driving the same state, but actually, according to the equation, that's just not possible. Like that's like so. If I drive my transitions in series, right? So here I have 1 transition. Here I have 2 transitions, but they're in series. Here I have 3 transitions and I have 4 transitions. I engineer these transitions so that they're in series, and I populate like each of the additional bands of the bound. But here, if I now add my transitions in parallel, so they're all just pointing to the same state, I thought like. Intuitively, I would have thought that I'm squeezing all the probabilities of the state. I should be able to get like a huge probability there, but actually no. Like all of these things collapse to because topologically the capital theta is only 1 theta for all of these. So the best you can do is just 1 theta. So like things are just like leaking out of this. So like here's the numerical results of that. So like sometimes in these kind of dynamic systems, just because of how much you're like depleting certain states, that your intuitive expectations of the outcome may not be what actually happens. Okay, so the second thing, so now we have that, so we use that, then we can prove the first thing that we actually ultimately care about, which is now, okay, I fix my theta and I go from some k prime to some other k. So let's say that my initially at equilibrium, right? When I go from k prime to k, I have this amplification. Now, if I'm not at equilibrium, the additional amplification I can get over this is bounded by 2 theta over kt. So if I have my equilibrium system and I change, if I mutate or if I add some ligands or whatever, and I get this much amplification, this is telling me that for the same system, if I drive it, I can get this much additional amplification. So now this is relevant for the case of, say, signaling, right? If I were to think about at equilibrium, how much can this signaling system do, right? That would be this ratio. And I say, okay, now I'm gonna apply theta. How much more can this signaling system do with the same k prime to k computation? It can do this much more. And so, I mean, this is a logarithm, so this is e to the 2 theta over kt times the amplification. So it's a lot. So that might get back to kind of one of your questions, like this is actually you can get a lot of bang for your buck. So you can actually get a lot of computational advantage from a certain amount of non-equilibrium driving. And so that's why maybe it doesn't have, like maybe this overhead is not as great, right? And then here's the result for if you care about the KL divergence between the two probability distributions. So if you initially had a k prime to k transition, and this is how much your probability distribution changed. Now you apply your theta, this is how much more your probability distribution can change. And notice that this one actually has 2 pieces, which means that if your equilibrium KL divergence is 0, you can still get a non-zero KL divergence. And this is actually taken advantage of in the cell all the time. This is, I mean, we're all familiar with the fact that at equilibrium, a catalyst cannot change the steady state outcome. A catalyst only makes a reaction faster. But not at equilibrium, a catalyst actually changes the steady state probability distribution, right? This is the action of a catalyst away from equilibrium. And then this part is how much you would amplify the equilibrium KL divergence. So even though this part, you have to depend on the equilibrium KL divergence being non-zero, the advantage of that is it gets exponentially amplified. So there's design kind of pros and cons. This is nice as you can use a catalyst, so you can use non-stoichiometric control. Like for example, many kinases are like two orders of magnitude less concentrated than the targets because they just act and then they act on another one. They're catalysts. They work here, so they have this advantage, but they only have a linear information gain. Here, you rely on stoichiometric control. So something that happens at equilibrium has to be stoichiometric. So you have something non-zero in the absence of theta, but the advantage of that is that you can amplify exponentially. So it's like different design constraints to think about. Okay, so here is what it looked like if we now just numerically simulate a lot of systems. So the purple is if we just put random parameters, and then the gray is if we do some optimization. And we're not actually sure how closely to the bound this goes because that's still we're still undergoing a numerical optimization. But unlike the previous bound, which is achieved very easily, this bound on the KL divergence may not even be tight because it's a constraint on the whole probability distribution. But it does seem to indicate that the bound is, even if it ultimately does not turn out to be tight, is constraining what you're allowed to do, right? So on the upside is how much additional information gain you have, and the downside is how much you can suppress noise. Okay. And we also numerically looked at the bound when you drive it farther and farther from equilibrium. So the dashed lines are the bound, and then these like different colors are our numerical samplings, right? And the bound does seem to constrain the behavior of the system, even if numerically we haven't yet gotten it to be tightly saturating the bound. Okay, and these are just some examples. If I look at this L example here, right? What L example is really looking at the linear part of the KL divergence, right? It's in this part, right? The exponential part takes off here. The linear part dominates here. So here, as expected at equilibrium, like the probability distribution from K to K prime doesn't change. The probability distributions are almost the same, but when you go away from equilibrium, the probability distribution does change and there's fluxes. But then when you pick an example from this part where the exponential part is starting to kick in, at equilibrium, there's some change, but that gets exponentially magnified or significantly magnified when you apply theta. All right, and the last part is just like, how does this manifest in, how am I doing on time? This is my my last slide. So okay. So how does this manifest in biological systems? So we just plugged in, so RAS is very well characterized, the RAS cycle is very well characterized. So we just plugged in the experimentally measured and inferred rate constants for this molecular machine. And here we're considering the K to K prime as basically there's these two things that are added. So what GEF does is it acts as a catalyst for these steps. So it's like helps GTP and GDP leave RAS, and then GAP it catalyzes the hydrolysis of GTP. And those are the two knobs that the cell can tune to alter the signaling state, which is this state. Like the probability of this state is the on state for RAS, which determines like cell differentiation and growth, et cetera. Like it's a master regulator, right? So this is the on state. And so in this case, we're just looking at the probability of the on state when we're in the case where the cell is applying GEF versus the probability on the on state when the cell is applying GAP. So these are the K and K prime states, right? And this is we're able to solve this numerically using the circuit thing we talked about. So this is the answer for the parameters of the system. And what's interesting is it actually is hitting the bound for theta, or I guess GTP concentrations below that of in vivo. So when I look at this, I infer it to mean that basically what happens is that once you can get your signaling ratio to be like on the order of 1,000, like you're on, like in this state, have a certain probability, and then in the other state, that probability increases, it gets multiplied by a factor of 1,000. That's enough signaling. So like beyond that, there's just like no more like need for that. But like I can imagine, evolutionarily, right? Maybe in a context way back when the ATP or GTP concentration of cell was much lower, right? So when theta was much lower, you would actually want to be saturating the bound because you're still not hitting the point where your signaling gets, for all functional purposes, maxed out. Like here, you want to hit like 100-fold rather than just 10-fold, right? So the fact that this is like a tight for lower values of theta. Oh, whose phone is this? Yeah. Okay. All right, and so we also apply this to microtubules. Again, we using the circuit formulas, and we can actually solve for the analytic steady state solution for this, which here has in principle an unlimited number of possible states, but we capped it at like an L max, maximum length, which is like the cell size effectively. Here, the K and K prime are basically tuning the rate constant of catastrophe. So which is something you can think of as a catalytic process. You're just changing the stability of the growing cap, and that determines the catastrophe rate. And it's a non-stoichiometric. It's a catalyst because once it happens, whatever modulating factor can go to another microtubule and initiate the catastrophe. Because once it's initiated, it banana peels the whole microtubule down, right? So you can't see the dashed line, but when theta is 0, this is 0 because this is the catalyst. So when theta is 0, the catastrophe rate constant does nothing. Okay, but when not at equilibrium, the catastrophe rate constant modulating that can do something, right? So we show it for what happens in terms of the probability distribution of microtubule, the ensemble of microtubule lengths. So here is a KL divergence between the probability distribution of lengths, right, in K, which is one value of the catastrophe parameter, versus K prime, another value of the catastrophe parameter. And so the length distribution gets significantly changed between, say, mitosis and interphase. But we also show what happens in the extreme case where we consider difference between like interphase and the limit where the catastrophe rate is so big that essentially all microtubules just fall apart, right? And in that case, you can actually get a very, very marked difference in the probability distributions between like the catastrophe, the interphase values of catastrophe and the super high values of catastrophe where basically microtubules can't form. Getting closer to that now. Okay, so yes, this is my last slide. This is like a summary. So is there some kind of thermodynamic law where we can use it to at least constrain our thinking of what's possible and what you can get? So in terms of the formulation that I laid out, right, of formulating in this way of how do amplification of individual states or the changing of the whole probability distributions depend on how strongly it's driven, like these are the results, right? This result, we've shown it to be numerically tight. This one, we haven't yet shown it to be numerically tight, it may not be, but it's still, as you can see from the data, pretty constraining on the KL divergence that's possible. Okay, I think that's all I have. This is basically, this work was just a long multi-year conversation between Eason and I. It's been really fun working on it with him, and Eason is awesome. And I thank thank you very much for listening and for the great questions.
Questions?
Yeah, I don't know.
So you mentioned how one of your results was related to like the Swinford's theorem. I'm wondering if there's any relation between that second result there and the like Kawasaki response relation, which is basically telling us that like if you have a distribution in or out of equilibrium, that's related to essentially like an average of an exponential of a work, conditioned on being in a specific state.
Sorry, can you say that one more time? I didn't hear the first.
So there's a response relation called the Kawasaki response relation, and it basically says that the non-equilibrium distribution is related to the equilibrium distribution by a factor that's like the Jarzynski equality factor. Only it's conditioned on ending at a certain state, namely the state that the probability distribution is evaluated at.
I see. So, but then, so, is it like, is it a relation for the steady state or is it for any time point?
Any time point.
Okay. So that, yeah, this is not really like a result that tells you about the time evolution of like a dynamical system. It just tells you what you can expect at the steady states, but it's something where it's in this case it's like something that's not like a perturbative response, but so you can think about it in terms of like what happens if you really impose very big changes between K and K prime or driving or not driving, but it is not something that's relating how you update like the dynamics of like an actual changing system. Yeah, so that's a big caveat here, is that this is only telling you about steady state probability distributions.
the fact that you are always looking at steady state. I was wondering if you try to compare, so in your talk, you always you always use the force as the measure of like thermodynamic gradient. I was wondering if you ever try to contrast this result with the entry production of steady state. For example, how the amount of information you can generate relates to the amount of entry production you spend to maintain steady state.
Yes, yeah, so I would like to do that. I just haven't succeeded in doing that. So like, I think one of the things though is that it has to be formulated in the, like certain things like dimensionally, like cannot be, right? So like here, right, right, so the theta is a thermodynamic force, so it has no temporal components, right? So as I showed before, the sum of the current everywhere times theta is the total rate of dissipation. But the current in there has a time dependence. And so fundamentally, you cannot relate information difference between before and after to something that has fundamentally a unit of time, which is the rate of entropy production.
Yeah, these analytically, but you have the computation, as you said, you can try to experiment around with them, no?
Yeah. I mean, I guess like, I mean, one other way to say what I'm saying is that you can imagine a case where you just multiply all rate constants by a factor of 10. So if you multiply all rate constants by a factor of 10, your equilibrium and non-equilibrium steady states will not be affected because you're just changing your time units, right? Theta is not going to be changed because it comes from ratios of rate constants. But your rate of entropy production is, because it is the product of theta times the net current. And now your net current has been multiplied by a factor of 10. This is what I mean by when I say units.